Welcome back to Your Brain, a Personal Tour. This is Steve Saltwick. Today's seminar is about the structure of memory. Memory is a slippery term. Some people use it one way, some people use it a different way. For today, we'll use a definition that memory is the storage and retrieval of knowledge about the world that changes our behavior. We'll look at memory in three different aspects. First, what are the critical parts in the brain that store memory? Second, do memories move around in the brain, in one part of the brain early on in your experience, and a different part of the brain later in your experience? And lastly, can we manipulate memory to relieve human suffering? My one-sentence summary of the seminar is that rich episodic memories Proust wrote about eventually become totem poles as the Pacific Northwest Indians carved. But the totem poles are in your brain. How does that work? Well, welcome to the seminar. Let's find out. Let's look again at the road less traveled metaphor we used last time. Seminar 2 was about how your brain decided to act, how to choose a road in the woods. Seminar 3 today is how does your brain remember what it chose? I've added a few more books to the reading list. They're excellent resources. However, they are academic publications, which means high prices. So be sure to evaluate used paperback or Kindle offerings. Now ready to begin? Let's go.
Ma chambre a la forme d'une cage. Le soleil passe son bras par la fenêtre. Les chasseurs à ma porte comme les petits soldats qui veulent me prendre.
Memories can be verbal or nonverbal. In some memories, we can be in a Proust story, a rich and detailed recollection of an event, perhaps long past. Or, in another memory, we might be in a Pacific Northwest Indian totem pole story. The memory reflects the gist of the story and is recreated anew with each telling or remembrance. Remember, a good example for a totem pole memory story for Goldilocks would be a bear, a bowl, and a bed. A knowledgeable storyteller, your brain, would recreate the story from these mnemonics. But there's a third kind of memory, and this memory is not verbal at all. It changes the brain structure that encodes an interaction with the world such that the behavior is different after that interaction. Look at this field. This field has previously interacted with water, which results in a gully. Further interactions with the water in this field will behave very differently because of this gully. This is a memory not involving words, but involving a change in the structure of the environment. I want to begin our discussion of memory with a personal encounter. This is me about to undergo my second hip operation. I'm accompanied by the brilliant surgeon, Dr. Shelby Carter. My first hip operation was extremely helpful. I just couldn't wait to have my second hip operation, to fully enjoy the benefits of having new hips and an old body. But this picture almost didn't happen, because a few days earlier, I had the following conversation with Becky. I was sitting at the breakfast table, drinking coffee, when Becky entered our kitchen, returning from work. She said, "Why aren't you at the doctor's office?" And I said, "What doctor's office?" Becky said, "To prepare for surgery." I said, "What surgery?" Becky said, now getting a little concerned, "For your other hip. It's time to do the right hip." I said, "I've never had hip surgery." Becky, now getting very concerned, goes, "Yes, you did, in March." I was very frustrated now, and I said, no, if I had hip surgery, I'd have a scar. And then I proceeded to show her how I had no scar, at which point I said, "Oh no, it looks like I have a scar." And then Becky goes, "It's time to take a little car ride." She didn't tell me we were going to the emergency room, but that's where we went. As we drove, it became clear that there were some things that were not quite working in my memory. First, I did not remember going to India 6 months before. Forgetting something within 6 months of this episode is significant. However, I did remember Becky and the dogs. I could remember the layout of her home of 20 years. I knew where to go find my shoes. I remembered how to put my seatbelt on. So I remembered the habits that one normally has. I could ask reasonable questions. For example, I knew that if I had a hip surgery, then I would have a scar. So I could do deductive reasoning. But I had a big problem, and that big problem was illustrated in the following dialogue we had as we drove along. Becky would ask, "How are you doing?" And I would say, "I had hip surgery?" "Yep, 3 months ago." I would say, "Where are we going?" And I had hip surgery? "Yep, look at your scar." "Where are we going? Really? You sure I had hip surgery?" "Yes, you had hip surgery." Now tell me again where we're going. I couldn't incorporate the context of the conversation, of what was happening right now, into my memory. I could focus on it for a little while, but then, distracted or looking at something else, I would return to the fact that I just could not represent that I had hip surgery. This leads us to look in some specific places for where my problem lay. The good news was that after a 24-hour battery of tests against my body, my brain, my heart, my blood flow, I got a diagnosis. And right then and there, after that, I got a new hip. And boy, am I happy to have those 2 hips. But what happened in my brain? I most likely had an interruption of blood flow from an internal jugular vein to that old friend of ours, the hippocampus. That interruption of blood flow acted as a reset. In essence, the interruption of blood flow caused my hippocampus to reboot. The medical condition is called transient global amnesia, TGA for short. My hippocampus had to restart, and as a function of that restarting, I had the memory episode. We'll use this story to set our framework for the seminar today. Why did a hiccup in my hippocampus result in a failure to retrieve memories? Why did it make me clang over and over again, to loop? I'll probably never recover the memory of the 4 to 6 hours of the actual episode, but soon thereafter, all the other memories, for example, the trip to India, came back to me. What's going on here? Now, if you talk about memory, there's a specific animal that often springs to mind: the elephant. Legendary for their memory, it does turn out that elephants have a hippocampus 40% bigger than a human hippocampus, prorated for body weight. It also turns out that elephants have one of the most complex societies on the planet. They comfort others, perform death rituals, and honor their elders. I wish more humans did that. The association of the hippocampus with a complex society is the first hint that there might be something more to the hippocampus than we studied in the previous seminar. And that's important, but there's also another good reason to be aware of elephants and the hippocampus. The elephant is only distantly related to humans. Our family tree diverged about 60 million years ago. Elephants are most closely related to manatees. So if elephants and manatees have large hippocampi, then the hippocampus should be fundamental building blocks of memory. For virtually all mammals, we can learn a lot from studying how the hippocampus works in such a widely diverse set of species, if we can keep them around. And help for keeping them around is one of the fortuitous outcomes of research on elephants. Nowadays, poaching is not the most pressing danger for elephants. Competition for forage with native farmers is the new challenge. Farmers often kill trespassers on their fields. I'm thrilled to note that as a result of some practical scientific research, it's been shown that elephants are truly scared of only 2 things: humans and bees. A bee can sting the end of the trunk, and that makes even a bull elephant cower. As a result of this research, enterprising farmers are now constructing low-cost bee fences, which are hives hung between poles with two outstanding results. Everyone can afford to protect their field without killing elephants, and the collected honey can be used for food and or a cash crop at the market. If that's not a win-win, I don't know what is. Recall, we looked at the hippocampus and found that there were unique neurons within the hippocampus called place cells. Those cells fired when an animal was at a particular set of space. Well, it turns out the hippocampus is much more than place cells. We'll look at that in this section of the seminar. To make a long story short, the hippocampus is essential for what is called episodic memory. Examples of episodic memory are: where were you when Neil Armstrong stepped on the moon? Where were you and what were you doing and who were you with on 9/11? Don't you recall? Those rich autobiographical memories, almost like a Proust story, are detailed, we remember them vividly, and as it turns out, the hippocampus is essential for this kind of memory. Episodic memory is not a YouTube video of the events of your day. The hippocampus seems to thread time and space into a personal fabric of experience, involving you at the center, and then the what, where, and when aspects fused into your experience into a unique way. While machines are used to measure linear time, see the clock markings in the middle of the diagram, our brain experiences time as an ordering of events. In other words, I went skiing downhill, I trudged up the hill, then I had to shovel snow and build a fire, etc. The metrics of this experience are far more complicated than in the picture. In fact, we're just starting to understand the experience in detail. But we do understand the basic points, and that's what I'm going to present today. I want to refresh your memory about place cells and how they relate to a rat thinking. When a rat runs through a familiar maze, he goes through a series of spatial locations. Those locations are coded in the hippocampus by different place cells. A place cell fires when the rat is in the purple zone. A different place cell fires for the yellow zone. A different place cell for the red zone, etc. All the way through the maze as he runs it. But there's another time that place cells fire. A sharp wave ripple. A sharp wave ripple is a very quick burst of place cells, about 1/10 of a second long, too fast for movement, and these place cells fire in sequence. So in essence, when a sharp wave ripple occurs, the rat is thinking about a path. He's not moving in that path. This happens much too quickly. The rat is thinking about going on that path. That path can be described by the sequence of place cells as they fire. On the bottom, a sequence of color-coded place cells within the shortwave ripple decode the trajectory in a maze. Go purple, blue, green, yellow, orange, red, and it takes the rat through the maze. Fantastic. But, and this is a big but, it turns out that that's not the only time hippocampal cells fire in sharp-wave ripples. For example, take a rat that's been trained to go through a maze to obtain a food reward. If we look at the sharp-wave ripple when the rat begins, we would see that the firing of place cells anticipate the path the rat is about to run. In other words, we can predict the path the rat will take to the food. But once the rat gets to the food and starts to consume, we see something very different. The place cells fire in the reverse order. It's as if the rat is thinking, how did I get here? That's probably a good thing to remember. The reward system is going to be active when the rat is consuming this food. So at the time the reward system is active, place cells are firing in reverse order in the sharp-wave ripple. They're basically laying down a path to this food that will be strengthened by the reward system. This mechanism is what underpins the deliberative learning we studied last seminar. This mechanism is very, very important. Place cells within sharp wave ripples show what is about to happen, in terms of the behavior of an animal, and they also explain how an animal learns when they discover a reward in a maze. And sharp-wave ripples could help the animal for a memory of what happened. Is that true in humans? Do we think like a rat? Well, you've heard me say it more than once. Yes, you think like a rat. There's recent research that shows this clearly. Here, patients undergoing surgery for epilepsy were shown pictures while they were on the operating table. Recall the violinist? There are no pain receptors in the brain, so implants that record brain activity are safe and pain-free. They were also asked to freely recall the pictures. In other words, they were asked to report out any picture they remembered without any explicit prompting on which picture. There were three major findings. 1. Strong rates of sharp-wave ripples were associated with stronger memories. High rates meant high rates of recall. 2. A sharp-wave ripple usually preceded the recall of a picture. In other words, the retrieval of a memory was preceded by a sharp-wave ripple. And finally, 3. The sharp-wave ripple in a free recall activated other areas of the brain in a manner and pattern similar to when the picture was actually viewed. The sharp-wave ripple brought forth brain patterns in recall that were like the patterns which occurred when the actual perception. The patients were, in a sense, seeing the object during recall, all driven by the sharp-wave ripple. Wow! These sharp-wave ripple dudes are really, really important in memory. But wait, there's more. It turns out that place cells also measure time. So some cells measure space, and some cells measure time. Let's take an example. Train a rat to run on a treadmill or in a maze for a varying length of time. If we look at the hippocampus, we'll find cells that measure time, much as other cells measure a spatial location. In this diagram, we see 4 hippocampal cells, A, B, C, and D. The activity of these cells seemed to measure time intervals, as a rat in this instance was on a treadmill. Cell A marks the beginning of the time interval, B the next, C the next, D marks the end. We show 3 separate runs on the treadmill, so there's 3 rows of each cell tracing. It also turns out that hippocampal place cells can be social and track the position of others. The coolest way I've found to show this is with an experiment with bats. Somehow, experimenters invented a way to record from bat hippocampal place cells while the bats were flying. Hippocampal, quote, place cells were found to represent not only the bat's own position in space, but other social place cells were found to represent a different bat's position in space. So if you're confused about why I'm calling cells that detect space, time, learning, and social elements place cells, I get it, I understand. Let's get a better name. Hippocampal cells certainly represent a lot more than place. What they're really doing is building a context. What do I mean by context? Well, let's take a familiar example. In an office, there's many different kinds of elements. There's a chair or chairs, there's a desk, there's a filing cabinet, there's a clock, there might be papers. All of those are individual perceivable objects. They're found in many different kinds of experiences: Amazon web pages, Costco, Office Depot, etc. In an office context, there's an integrated whole of them in a particular place at a particular time. So your office is a particular desk with a particular filing cabinet, a particular chair, particular clock, particular papers, all integrated into a perceptual whole. That's what I mean by a context. And that context has all the details of a Proust story. It's rich. You can imagine yourself within that context. The essential function of the hippocampus is to encode space, time, learning, and social variables into such a context. Since that context can be used to affect behavior, which is our definitive memory, there's at least one type of memory processed and held by the hippocampus. Once context cells are built in your hippocampus, they can be associated with other experiences. If you have an aversive experience in that office, let's say a confrontation with an office worker, then that office context can be associated with that averse experience, as if it's Pavlovian or associative conditioning. An interesting aspect of context cells is that their response can be evoked by just parts of the context. It's called pattern completion. When you remember or experience just a portion of that office, all the other memories associated with that office can be called to the fore of your brain. The presence of the office can bring forth an aversive emotional response even with the person absent. The context is being completed by the hippocampus into the whole office, including the memory of the aversive experience. A good way to think about the action of the hippocampus, then, is that it helps your brain create a movie of the experience. It's trying together who, what, where, and when of the experience. And this movie is centered around you and your experience. However, the movie is a special kind of movie. Your experiences are abstracted to shareable units of knowledge. Now let's take an example, a meal. In a typical American meal, you might experience a sequence like this: an appetizer followed by a main course, and then a dessert, why not? And then coffee. What your brain does is to not only make a movie about it, it abstracts the experience into units of knowledge that represent key dimensions of each stage. Appetizers are small and often shared, and savory, in other words, not sweet. Mains are large, especially in America, not shared and varied. Desserts are small, often too small, and often shared and sweet. And then finally, since I'm an exclusive coffee drinker, the final course is coffee and not tea. Recall the seminar title, This is My Personal Tour. Before we close this section, I want to return to the fundamental importance of place in the operation of the hippocampus. Yes, the hippocampus reacts to far more than just GPS position, but also how they are organized. The most important recent research would say a cognitive map is being constructed. In this view, the context is really a map of your sequential experiences in that context. Let's take a simple view from a rat's perspective. The rat explores a cage with various colored walls and objects. It's proposed that the hippocampus is building not only a movie of the sequential experience, walk past a red wall, walk past a red wall, walk to where the red wall meets a black wall, turn, encounter a light blue object, and then hit the dark blue wall. The rat is building an actual model of the context and environment, shown in this panel as a map. Allocentric means it's just not centered around the rat. It's a map of the space. This is right in line with how our sensory systems build an object and the act of perception. It's an exciting theory, and it'll be fascinating to see how further studies advance our knowledge. So let's hear from Homer. The first thing Homer would say is, wow, place cells are really context cells, not only for the perception of context, but also the memory of context. Many of the same cells that perceive a context are the ones that are associated with the memory of that context. The second thing Homer says is his brain makes a movie about experiences to help the formation of a memory for that experience. In The Tempest, Shakespeare says, "We are such stuff as dreams are made on, and our little life is rounded with a sleep." Shakespeare got it right. The creation of memories depends upon sleep. Human life would be impossible without sleep. Your brain needs sleep, and we'll talk about that in this section. Dreaming is an activity of your brain during sleep. It's not essential. Certain antipsychotic drugs eliminate dreaming, and people still live healthy lives. But dreaming is something that has fascinated man for as long as history, and we need to talk about it in our search for memories. Sleep is divided into two main categories as shown on this diagram. There's something called non-rapid eye movement sleep, or NREM, and something called rapid eye movement sleep, or REM. The various stages of sleep within these categories are shown. Sensation and perception varies across these modes of sleep. Most people think that dreaming only occurs during REM sleep, and until quite recently, that was supported by the output of research literature. But as it turns out, as so often with research, the result you get depends on the question you ask. If you ask people going into the first stage of non-REM sleep, anything going through your mind, you will find you get a dream report 80 to 90% of the time. If you wake people up in a stage of deep sleep, there's something called sleep inertia, and it takes a major effort on the dream, on the sleeper to produce a dream report. People awaking from this stage of sleep need time to reorient and to get back up to speed on what's happening in the now. Dream reports are very unlikely at this point in time. But if you sum up over all the stages of non-REM sleep, dreaming is reported about 25 to 75% of the time. Clearly, we dream in almost all stages of sleep. If you look at rapid eye movement sleep, REM sleep, it turns out that the vast majority of rapid eye movement sleep occurs in the womb. A baby before birth will spend up to 24 hours a day in rapid eye movement sleep. That sleep percentage declines over time, becoming almost vestige-like once we reach adulthood. A fascinating theory of REM sleep function, at least in the womb, is that during REM, a pattern generator in the brain causes the baby's limb to move. That movement then feeds back via somatosensory input into the brain and helps various parts of the brain wire together. This pattern generator doesn't go away as we age, so there's still bits of our sleep where this pattern generator is generating movements, and that may cause our body to move, and it may be what's causing active, lucid dreaming. One thing that sleep has unambiguously been related to is the reduction of synapses between brain cells. New synapses are essential to learning. New ones grow as we make new memories. But wait, brains are typically in skulls of fixed volume. If you make new synapses every time you make a new memory, won't we run out of room? Yep, we will. So how does Mother Nature address that? Sleep. Sleep reduces the number of synapses in your brain. In essence, sleep helps you forget. Research confirms that this happens in a wide variety of animals, as diverse as fruit flies and mice. Actual micrographic photographs of synapse reduction prove this. There seems to be a grand total, that is kind of like a maximum number of synapses that a brain can have. Sleep reduces the net number of synapses in your brain to relieve the pressure of daily learning. Now that's an interesting fact, but there's even more interesting facts that can be discovered about the structure of a memory in your brain, and let's look at that next. Sharp-wave ripples are repeated in sleep, slow-wave sleep, the sleep that occurs in the first half of a night rest period. This occurs virtually in all mammals. This phase is called slow-wave sleep or non-rapid eye movement sleep. And in that, we see representation of thoughts being repeated in sleep. That keeps memories from being forgotten as a result of synaptic reduction. If we query humans about what was going on in their minds during sleep, we see a clear pattern. Recent memories are reported in the early parts of the night, and remote memories are reported late in the night. Since sharp-wave ripples are most likely to happen in the first half of the night, I assert that sharp-wave ripples are working on our most recent memories. I think they are transforming the memories. But before we get to that, let's make sure our assumption that sharp-wave ripples are fundamental to the establishment of memories is solid. So let's set up an experiment. Our goal is to create a memory, because if we can better understand the brain processes that underlie it. First, we're going to take a rat and we're going to put an electrode in his hippocampus, and we're going to record place cell activity and sharp wave ripples. We're also going to put an electrode into the reward system. By stimulating the reward system, we can behave as if we fed the animal some food or given him some kind of positive reinforcement. We find a place cell in this instance, and the place cell is shown by the red dot of activity. We're now going to let the rat go to sleep, and we're going to monitor his hippocampus. Whenever that place cell fires, we're going to stimulate his reward system. In this phase, the rat is asleep. He's not moving around, it's as if he's dreaming about moving around, and whenever he dreams that he's in that location represented by this place cell, we're going to stimulate the reward system. After the rat wakes up, we're going to put him back in the environment, and lo and behold, the rat makes a beeline for that area of the maze associated with the place cell. Amazing. We have created a memory. The rat never experienced food or any other reinforcement in that place. We've done this all electronically. We monitored the place cell of the hippocampus, stimulated the reward system whenever that place cell fired during sleep. And if we can create memories by pairing sharp wave ripples in the hippocampus with stimulation of the reward system, can we do the opposite? Can we disrupt learning and memory by disrupting sharp wave ripples? And it turns out we can. In this experiment, there's an 8-arm maze. A rat is always put into the middle of the maze to start. 3 out of the 8 arms are baited with food. Those are shown by the small red dots at the end of the arms. Each day we give the rat 3 training sessions. He's placed in the center. He gets to explore and find the 3 places of food. He gets 3 trials a day. There's a 3-minute rest in between each trial, and then at the end of 3 trials, there's a 1-hour sleep rest period. During that sleep rest period, we're going to monitor for sharp wave ripples. For a test group, whenever we see a sharp wave ripple, we're going to disrupt them by zapping the hippocampus. In a control group, we'll zap the hippocampus the same number of times, but we'll do so randomly with respect to sharp wave ripples. The test group has a severe impairment on this task. By disrupting sharp wave ripples in the hippocampus, we are disrupting learning and we are disrupting memory. So we can manipulate memory. We can actually create them, and we can actually disrupt them by manipulating sharp wave ripples. I think our assumption that sharp wave ripples underpin memory is solid. We're now ready to explore how memories move around and are changed in our brain as a function of sleep. The bottom line here is that sleep produces memory totem poles. Remember, totem poles represent the essence of a story. They're the distilled elements separate from all the autobiographical memories. In this section, we'll see how sleep produces memories like totem poles as those memories move outside the hippocampus. A good experimental protocol to show the movement of memories is the social transmission of food preference. Now, that's a mouthful, but it makes a lot of sense if you think about it from a rat's perspective. In this kind of experiment, we take 2 rats, one's called an observer, one's called a demonstrator. We ensure they trust one another, if you will, by having them share a meal. They eat some well-known food like rat chow together. After that, the rat are separated. The observer goes away into a completely different room. The demonstrator is then given some uniquely flavored food. After the demonstrator has eaten this food, say the cinnamon, we bring the observer rat back together with a demonstrator and they socially interact. When rats do that, they smell each other's breath. After that reunion, we present the observer rat a food choice. One choice is cinnamon, which the demonstrator rat has eaten and the observer rat has smelled on his breath. The other choice is cocoa rat chow, which neither rat has eaten. When presented this choice, The observer rat shows an incredible preference for the cinnamon-flavored food. This makes a lot of sense. If you smell unique food on the breath of one of your rat pals, and he's not dead, well, that's probably a pretty good food to eat, and a good memory to have. Let's track the development and movement of such a memory over sleep time. We're going to see which areas of the brain are involved over the course of time, and what we find is, early, say in the first couple of weeks, there's an interaction between the hippocampus and the frontal cortex of the brain. There's a synchronization, if you will, between the hippocampus and the frontal cortex that establishes this early memory. Both areas of the brain are required for this to happen. If the hippocampus wasn't there, there wouldn't be retention of the memory. If the frontal cortex was removed, there wouldn't be retention of the memory. You need both. But then later on, the memory is in the frontal cortex only. It has moved and been transformed. That memory now isn't in the hippocampus, it's in the frontal cortex only. The hippocampus could be inactivated, much like mine was on that fateful day, and the memory would still remain. So here's a good example of how the passage of time with sleep causes a memory to be consolidated outside the hippocampus into other cortical areas of the brain. That's extremely important. That's the reason why I remembered my wife Becky, our beautiful dogs, even when my hippocampus was restarting. Now, before we finish this section, I want to look at one more detail of the memory as it synchronizes. Because it's very, very important to see how memories not only can move about, but how they can be transformed. In this experiment, we challenge human subjects with a fairly complicated mathematical task. We don't need to be concerned with the details of the arithmetic, but what we really need to understand is that there was a trick. There was a hidden pattern in this numerical task such that the final answer was predicted by the second number in the sequence. None of the subjects were told this. They were given a fairly complex numerical task to do, then they had to proceed through the task. Some were allowed then to sleep on it, others were not. Then they were tested. A main point is nobody was told there was a shortcut to solve this challenge. Now, if the subjects were allowed to sleep, 60% of the subjects reported that they gained insight and found the shortcut. This result is outstanding. It says that memories, as they consolidated, are subject to abstraction. Hidden rules or patterns are brought out when the hippocampus and the frontal cortex interact. So if your grandkids are cramming for tests, you might want to tell them, get some sleep, because neuroscience says they'll be amazed at the kinds of insights and observations their brains can come to when you just let your brain sleep. Let's try to tie all of this together using as an example the best way to squeeze toothpaste. Early in the stages of memory formation, the interaction has the characteristics of a Proust story. It's rich, it's detailed, it's autobiographical. You are remembering the sequence of things that happen to you. The hippocampus deals not only with the frontal cortex, but also several other areas of your brain. It's synchronizing, talking, if you will, to several different areas of your cortex during the creation of the memory. As sleep occurs, the memory is consolidated. It moves to other parts of your brain and transforms as if into a totem pole. Elements are abstracted, rules are evaluated, insight is gained. The autobiographical, the you part of the memory, may well decrease. The memory goes from a Proust story to a totem pole abstraction squeezed from the bottom. Abstraction has evolutionary benefit. It gives you the rules of the road in much less space. And if you're going to learn new stuff, you need space in your brain. Additionally, by moving the memory to many different areas of the cortex, you protect the memory. The hippocampus could go haywire, but you'd still retain the memory/totem pole. In other words, Proust has become a totem pole. Let's get Homer's take. Homer's ecstatic. He says sleep is important to form long-term memories. Yay, sleep. Homer, as you well know, loves to sleep. Now he's going to think that his sleep is justified by neuroscience. Homer has an additional point, and that is the hippocampus plus cortex interaction creates totem poles. It abstracts information. It creates insightful observation, depending upon the capabilities of the animal brain. I think Homer's pretty well nailed it. These are the amazing properties of sleep and the formation of memory. In our last section, we'll look at memory manipulation. Is it possible to alter or modify memories? And would that be a good or ethical thing? I think we can agree that there's some type of manipulations that are not ethical. Erasing the memory of an innocent bystander, for example. Mother Nature gives us memory to remember unpleasant situations for a good reason, so that we don't repeat them. However, if we have the ability to alter the emotional content of a memory, would that be a good thing if it improved the life of an individual? As an example, let's take anxiety disorders. These disorders are very common. They affect millions of lives in the United States and all around the world. They reflect intense emotional dysfunction, from soldiers returning from a tour of duty, irrational fears of animals or of heights, dysfunction associated with abusive relationships, or instances of rape. 30% of our spending in the United States goes to some form of anxiety disorders. The cost to the economy is significant. Over $45 billion are spent trying to alleviate the suffering that comes from anxiety disorders each year. If we could separate the emotional dysfunction from those memories, would that not be a good thing? Would that not be a good thing to use the technology and understanding we've investigated today? Let's take a look. The example I want to use is PTSD, post-traumatic stress disorder. It occurs in soldiers returning from a tour of duty, and it's thought to dramatically involve overgeneralization. By that I mean there's a memory of a combat experience which has a significant sensory element, let's say some kind of burning wreckage. After returning home, the soldier experiences something that's similar, like a backyard fire pit, and there's an overgeneralization. That causes the recall of the combat experiment. We've seen how the hippocampus can jump to conclusions and perform something called pattern completion. Well, this is an example of that. In PTSD, the hippocampus cannot discriminate between the two sensory experiences, and the former soldier is thrust back into the intense emotion of combat experience by an inoffensive cue. Someone with a healthy hippocampus, the pattern discrimination can separate those two environments. The backyard fire pit is a context separate from the combat experience. In this former soldier, that's not overgeneralization. There's not the intense emotional dysfunction associated with combat with everyday experiences like the backyard fire pit. How can we go about relieving the symptoms of this debilitating anxiety disorder? Well, it turns out one way to do this is to grow new cells in the hippocampus. In a normal hippocampus, we do grow new nerve cells throughout the course of our life, about 40,000 a day. Sounds like a lot, right? But that's not that much when you've got 86 billion already there. It's called neurogenesis, and it occurs in a very few parts of the brain. However, stress has an adverse effect on the growth of these new cells. Combat experience is the textbook example of stress. So from exposure to combat, we probably have a decline in the growth of new cells. But many events can increase the growth of new cells in the hippocampus. The passage of time, certain kinds of anti-anxiety drugs, electroconvulsive shock, exercise and meditation can all have positive effects on the growth of new cells within the hippocampus. This is a new insight and originates from research completed quite recently. The research extends our understanding of how PTSD involves a lack of discrimination of context, and it gives us hope that with various avenues of treatment, we can alleviate the emotional dysfunction that comes from this anxiety disorder. There are also new therapeutic avenues for treating anxiety disorders, which utilize the fact that the hippocampus is always learning. Because of that fact, we can combine non-fear memories with fear memories of the past to reduce emotional content. For example, a soldier suffering from PTSD can recollect fear memories in an environment of measured breathing, of general wellness of the body. As this new memory is consolidated, those fear memories are going to be intermixed with non-fear memories. That means we can use inexpensive, non-invasive techniques, not drugs, not electroconvulsive shock. We can reduce future anxiety from fear memories by coupling them with non-fear memories. This is a wonderful avenue of therapeutic approach. It deals with meditation, it deals with exercise, and it's an exciting area of research that we'll hear more and more about in the future. But it also has implications that a memory is really the last retrieval of an event, not the original event. If I'm asked to recollect an event that happened long ago, I'm not going to get the original memory of the event. I'm going to get the memory as it's been recollected over time and coupled with all the other context when I was recollecting. That has implications for eyewitness testimony, especially as it relates to events far away in time. Our legal system is grappling with such effects. Neuroscience can inform those discussions, but new legal scenarios need to be envisioned for what we now know to be the complex dimensions of memory and its transformation in your brain. Last time for Homer today. Homer says, your mom was right. You should face your fears with support. By bringing up your fears in a safe, non-fear environment, one can alter the composition of a context of a memory. That's a potent way to assist addressing disorders. This technique is improved with meditation and exercise. I think it's important that as we deal with anxiety disorders and other syndromes affecting a large portion of society. On this point, Homer would also like to say, why don't you also try it with exercise and meditation? It's amazing the amount of scientific data that is coming out in support of exercise and meditation for their beneficial effects on the hippocampus and on the brain in general. In fact, on the body in general. We'll look at this in more detail in Seminar 6. This concludes Seminar 3, The Structure of Memory. I hope you enjoyed the seminar. In Seminar 4, we'll begin to look at the question, what's really so unique about human brains? Up till now, we've talked about basic brain processes similar among animals of the mammal world. In Seminars 4 and 5, we'll explore the uniqueness of the human brain when compared to other primate species. We'll see some fairly surprising results. For example, how the search for bananas in the forests of Africa set the shape of your brain. That brain shape set the stage for the right-hand part of this diagram, a sculpture of the itinerant Japanese Kuya, who lived in the 10th century. No animal other than a human could produce the right-hand part of this slide, but no human would have emerged without the left-hand part of this slide. I look forward to you joining me on this fascinating journey. This is Steve Saltwick.